Just waiting for the seconds. There we go. 10 a.m.
Yep.
Yeah, Nicholas, you want to? I can jump right into it if you give me a little intro.
Yeah. So Colin, you're the first one out today with Wetlands in Space. I'm thinking Muppets there, but you're not.
No, that was kind of the joke, actually. So you got it. It was pigs in space. Anybody remembers that from the 80s there?
So self-organizing wetland bioreactors, sowbs, for biological lunar regolith processing and cycling, which Colin has been doing for forever, I was about to say, but 15 years at least.
Yeah.
Well, the floor is yours.
Okay, great. Hey, Stuart. Yeah, so my name's Colin Lennox. I'm the biofilm technologist for EcoIslands LLC. I'm also the CEO, but I'm an SME for bioreactors and the like. I also work with Arcology Solutions, of which Stuart Nelson is one of the founders. He just popped in here. And I'm going to be talking a little bit about what I do, why the science is important, but then also I'm going to do kind of a brief pitch for the rest of Arcology Solutions, because please join us. Most of our members are here at this conference, actually, and Nicholas shows up every week as well. And thank you for that, Nicholas. So self-organizing wetland bioreactors. So I build wetlands in a box. They used to be in boxes. Now they're in pipes. And there's a picture of me. This is in Livingston working with Stuart to build one of the, I think that one's just a cistern, but it just kind of demonstrates some of the techniques and technologies we're using for extrusion welding. And this is just a long process to get at essentially how do you make acid mine drainage useful? AMD is from mining impacts. And then most of my clients then are from mine drainage, reclamation, coal mining, or hard rock. Anyway, okay, so what is a wetland? Because essentially we're working with wetlands, and generally this is an engineering heavy field that we're all in. And so there's not a whole lot of biological background generally. So that's why we're just gonna kind of start from fresh. So what is a wetland? Holds water most of the time, moves water through it, or water moves through it, high surface area to volume, high biomass to volume. Cycles matter and energy lots of different ways, biotically and abiotically, and self-organizes by Gibbs free energy and the prevailing atmosphere. And it's okay to be scratching your head. We'll get into that. But just, those are my definition of what a wetland is, a naturally occurring wetland. Okay, and then what is a bioreactor? It does predictable and reproducible work. Once again, these are my definitions. It is a biological engine ruled by the conservation of mass and energy, catalyzes reactions, producing useful products. So this is BISRU. Instead of in situ resource utilization, it's biological in situ resource utilization. Natural wetlands are self-selecting and organizing, which is also generally called natural attenuation. If you got an impact coming in, there's a, say you've got a bunch of iron coming in from a mine seep into a river. You would put in either, you'd put in a constructed or manufactured wetland to capture that iron and deal with some of the other impacts that are also associated with that, like pH drops or increases in acidity and the like, that then leaches other metals out of the ground and then produces this a whole bunch of impacts combined. So you could have like iron and aluminum and manganese and high acidity, high sulfate, lack of alkalinity because it's all getting used up, lack of oxygen in some cases. So natural attenuation is to deal with those impacts. But do you want to take up 10 miles of your river bed to deal with those impacts, or do you want to squeeze it in one tiny little area, get it all taken care of, and then the river is fine? So the constructed and or manufactured wetlands are giving designers and other engineers and biologists an opportunity to take care of those impacts long before they get to the river. But usually that's in a heterogeneous muck. A lot of times what's being produced is just, land application at best. It's not really useful. And that's where a manufactured wetland comes in. So moving forward, but also bioreactors specific and by definition. And once again, these are my definitions. Natural wetland Methane digesters, it's a batch reactor. I build flow-through reactors. Reclamation ponds are flow-through reactors. Fish tanks, batch reactor. Greenhouses, technically it's you could consider that one kind of a flow-through, but you can see where the lines begin to blur a little. Wastewater treatment plants, wine and beer vats would be batch reactors, but it's still a biological reactor towards a purpose, producing ethanol. People, plants, box turtles in my dog river there. If we are the classic Star Trek: The Next Generation episode with the crystalline entity called us bags of mostly water, something along those lines, that reproduce. And, whether we have a purpose or not, we certainly do seem to get a lot of work done. Whether it's useful or not is another question. Biofilm. Okay, so within every wetland is an enormous amount of biofilm. That's where I said that high biomass to volume. And biofilm is a mix of microbes and they're secreted colloids. Colloid is like mayonnaise, and remains generally stable in mass, population diversity, and density. So you could have, hundreds of thousands of different species within, one Petri plate of water that you pulled up, say, from something like this mine drainage site here. This is a raw seep coming out of a ground where there was a borehole dug to empty a mine, so it wouldn't fill up. So that's, the worst kind of case scenario. And it is right next to the stream too. This one is really difficult to deal with, and it has a high flow. It's about an hour from my house, an hour and a half. Main components, but most importantly, this biofilm then. And when you wake up in the morning and scrape that stuff off your teeth from brushing your teeth, that's biofilm. Covers every square inch of the planet. It's considered it might be up to 50% of all the biomass on the planet. We just don't see it. A bunch of it's up in the atmosphere. A whole bunch of it's going deep, deep, deep into the ground. There's all different kinds of metabolisms involved also with biofilm, from deep sea thermal vents to plants, to you and I breathing oxygen right now, just like fungi, to the other major kingdoms like bacteria and archaea. Excuse me, getting over a head cold. But the point of this, the film, then, is that the main components it protects the microbes producing the film, making them stable, and without getting into too much more, that's just essentially allowing them to persist and also deal with all sorts of impacts, and not to just be immediately wiped out because they're microbes. Main components in natural cycling, it's a media for chemical signaling, structural support, yada, yada, you get the idea. Won't get too much into this one either, but the redox ladder, here, I'm going to jump too forward. All right, so redox ladder, this one's written for the engineers. So these sowbs are bioreactors, and they do work. And they can take a whole bunch of different materials and separate them, essentially. And I'll go back, but just real quick, just so in this context and application. So sowbs are essentially multi-fuel and sowbs, self-organizing wetland bioreactors. That's just the term that I use for these organizations, or for these units. Multi-fuel and multi-oxidant biological engines that self-organize to use each reductant and oxidizer sequentially based on the energy released and the availability of the metabolic pathways. What does that mean? Essentially, it's a biological distillation system. Little piece of history. So the guy who first figured this out is named Sergei Winogradsky. He was a Ukrainian scientist and an early microbiologist. He figured out the nitrogen cycle. That's really important. But he also figured out that if you take a bunch of pond muck and you put it into a column, that it'll self-stratify. So that homogeneous mix of all the different muck from just some pond muck in any old stream, river, wetland, he pulled some of that material, and it turns out that it then will self-organize based off of those energy, the Gibbs free energy. So just to give you a little bit of context and like a visualization. So where you see here are a couple of diagrams of Winogradsky columns and how they start off mixed, and then they will self-organize. The units that I then build, go forward just to give you a little bit more context. So the units that I then build and put in on mining sites are a Winogradsky column put on its side, so that the areas as there are changes in the load, which is the concentration times the flow, or the different kinds of impacts in a water source, can then be divided by their availability and their usefulness to different microbes, depending on how much energy you can get out of that particular metabolic pathway. Once again, fungi, archaea, bacteria. So you're thinking sulfate reduction, you're thinking iron oxidation or reduction. Without getting too much more into it, geez, fine. So that's essentially, without getting really super deep into what's called the redox ladder. So you had mentioned that there is a stratification and that there is a semi-distillation effect. That's pretty much it right there, looking at that the pyramid there, where you're starting with aerobic conditions at the top and methanic conditions at the bottom. Now, these are measured by what's called ORP, or oxidation reduction potential, or an ORP probe. It's like $20 online. I mean, this is a ubiquitous tool used in, more specifically, the field of study is primarily called limnology, or the study of terrestrial freshwater environments. The other side of that would be oceanography. Okay, so whether it be marine or freshwater, determining like lake characteristics. That's broadly the discipline that something like this, and then acid mine drainage is within. So if you're like drawing a Venn diagram, you got AMD there in the middle, then it'd be a mixture of a couple different disciplines. So why it's important here for the redox ladder is, in normal environments, really the only impacts that you got to worry about. And when I say normal environments, like a river with too much nutrients in it, nitrogen, potassium, phosphorus, nutrient salts. But if you're talking about, or if you're talking then about a wastewater treatment plant, sewerage, or a biological life support system, now you got to worry about things like heavy metals that we humans ingest and then excrete, fecal coliforms. There is CBOD, which is chemical and biological oxygen demand. We breathe out most of our CO2 that's coming in. There's not a whole lot left over in our waste, but there's still some, and that's called BOD, and that's where fecal coliforms are coming from. Urea from urine. These are all different impacts that you would see in wastewater treatment that will have to be dealt with for Beckless or a biological life support system. And Frida's talk next is even more in depth, so I'm going to leave that there. But then specifically within the mining, you're talking iron, aluminum, manganese, selenium, sulfate, some of the real nasty stuff like uranium, cadmium, mercury, lead, tin, zinc. These are all the different potential impacts that come from active mining processes, and then we'll get into the water. It's particularly bad here in Pennsylvania and the Appalachian chain, because we've got that acidified mine drainage, where hard rock mining usually won't spool itself up and out of control. Acid mine drainage is where it grabs a hold and it just gets worse and worse and worse, because it's an acid producing process that then dissolves all the rest of the metals within that particular load that are in contact with that highly acidic water, which normally is awful. We don't really want that. But there are opportunities then when you have a system that is capable of biological distillation, and you know where the redox points are, and you've built a system that's large enough to be able to capture and maintain that stuff. When I say maintained, there's three ways that different elements are coming out of one of these sowbs. It's either through sequestration of an oxide or a sulfide, voltization of a gas like hydrogen sulfide or CO2 or methane, or ammonium, NH4. Or as a salt. As a nutrient salt, either like an actual salt and they're miscible. So it's easier to get the water out of the salt than the salt out of the water is a way to think of it. And so like potassium and phosphorus, sodium and chloride, lithium, fluorine, fluoride, depending on what its oxidative state is. But looking here, so you've got oxygen, reduction with water and in reverse. So that's electrolysis essentially. And electrolysis works well with these systems when using carbon anodes or electrodes. But then you've got nitrogen species, nitrate, and then manganese species, species, manganese oxides, and that's what my primary research is in, manganese and rare earth elements. That's, to me, that's the most, it's the hardest and it's the most interesting. You get that one, everything else is easy by comparison. We're still working on that one, to be honest. And then iron, sulfate, selenium is in between iron and sulfate. It's just not on this list because the sulfate's the big one. Then carbon dioxide reduction to methane, and then oxygen or water reduction to hydrogen, H2 there. That's generally a little beyond anything. I've never worked down to that level personally. And the voltage entirely for that system is 0.9 millivolts in this case. It's a 450 DC, and this is a DC based system then, if you think an AC versus DC. Okay, so that's how, where, and why it self-organizes. This is something from mine drainage. I'm not going to get into this one. I published this one. I'm happy to share it with you later on. Trophism, not getting into that one, but just to show some pictures. There's Stuart with one of the units. That's a wastewater treatment unit up in Livingston. Still looking for a place to home that one as we build our the arcology there in Livingston, Montana. More of the units. This system, unfortunately, was not successful. The bottom 8 units there, the ones on the bottom left, weren't able to pull manganese. And we're still trying to figure that one out. Some sites, it works like gangbusters. And I believe we have a world record for biotic removal of manganese as an oxide. You know, it's been quantified by Penn State, the Smithsonian. It's down at Oak Ridge National Lab. You know, so we know what we got out of it. We know under what the wet water conditions were. We're just still not entirely sure what the, you know, whether it's a biotic or an abiotic factor that is removing the manganese from solution and precipitating material that's 85% pure. Normally we get it 60% out of the ground from mining from China, and we're going to get out at higher rates. Now, why it didn't work here and will work on other spots, I've got some theories. Well, I mean, I've got hypotheses that could have to do with, you know, maybe there just wasn't enough manganese in the water and that you have to hit a certain rate for the catalyzation of the biotic process. That's one of the standing ones. We also know things like iron has to be down below 0.35 milligrams per liter. Having, what was it? Strontium in the water may act as a catalyst. I'm not entirely sure. So we're still working on it. But yeah, so this system got pulled, but the top end system there on the right worked just great for removing aluminum. And then it was able to keep the limestone bed directly after it clean, much cleaner than normal. Standard systems don't have that kind of pre-polishing generally. So here's some other units we built. These were for wastewater treatment from a mushroom production facility. We got inundated by too much oil and grease from their fryers. So this system was taken offline until they get that taken care of. And now they're just sitting on their hands because that's business. Let's see, here's my dog again. Some smaller wastewater treatment units that I built and leading to, well, no matter what you do, you still need stink pipes. So that's one of the other things we're working on. There's that 85% pure manganese oxide. So that came out of the units, and then we just knocked the material loose, gathered it, and there you go. That's where you would find your rare earth elements too, if there were any in that site. For lunar or Martian settlements, then okay. This is something that was put together by myself and some others for the Mars Society. One of their challenges about 5-6 years ago. The blue squares, rectangles, whatever in the middle there, the 3s are the sowbs, and the 3A, 3B, 3C, all the way down to the 3I, whatever plus. That's the redox ladder that I was talking about, just with some more detail, and then specifically what you would get out of what useful products. Hydrogen sulfide. You know, how do you make caustic soda from your perchlorate? Say if it's on Mars. Obviously, this is the Lunar Society, but you know, these are still this is quite likely to be a system that'll be useful pretty much anywhere you go. 0G, 1/3, 1/6, full G. You know, technically, I don't think that it should really matter as long as you have the microbes with you, because you know, want to go back to that, circle back around. As long as there is the presence of the different metabolic pathways that are required for whatever the impact is. If they don't exist, you're probably not going to work. So there's always caveats to everything. Ways that you might be able to replace that or take more with you. CRISPR-Cas9, not really sure. That's not my field. I'm essentially an ecologist. I'm not that. That's not my bag. But there are ways potentially around that with technologies that are coming out now. But most importantly, here's what I want to point out is that the ones there, this is regolith processing. Like I said, this is for Mars, so 2A, 2B, and 2C. Those are all perchlorate and other salts. So just in practice, however you gather up the material and then do your initial processing, first processing in a system like this is going to be water-based. This is a very water-intensive system. It produces a lot of different byproducts, but ups and downs. So I'm just this is not a panacea or a panacea here. But when used in conjunction with other technologies, it would provide novel products and save an enormous amount of energy in big scale. Because, we don't have an atomic wrench, as Quinn, a friend of ours, has said before. I like that term. Just because you can do it, you're still limited by energy. So you still want to do everything you can to get the most efficiency out of what energy you are putting into a system. But the basic idea, like going back to this, so 4A, 4B, 4C, those are your HABs. 4D and 4E, manufacturing, industrial. Once again, this would be some kind of a habitat on some other body of, Mars or the moon. And the idea being here that the carbon that you've got, you have to continue to recycle that through your habitats. But if you're using a biotic system to do the bulk of your processing for your regolith, then you need to incorporate material, carbon, back into that process. So you're essentially using your waste products to continuously incorporate new regolith into your systems. On Mars, obviously, that's less of an issue because you've got not a lot of CO2, but you still have some. But on the moon, I leave that one to smarter people. That's a question that I haven't spent a whole lot of time on, but just trying to point out the pitfalls in something like this as well as the opportunities, to be fair. So basically this using the wetlands as the great cycler, like they are here on Earth, may give us the opportunity to actually make it. Sure, we can get there, but are we going to stay there? How do we make sure that we're recycling everything, and in such a way that we can use our waste products to, I'm not making any claims on exponential growth due to regolith processing, but this could be a way to get towards that. Okay, and then I wanted to go back just real quick. So I got a couple minutes. Yeah, still got a couple minutes. So this was a presentation overall, kind of see me jumping around here. This is for the Analog Astronaut presentation that I did here down at Biosphere 2 earlier this year. And it was also to kind of introduce Arcology Solutions, of which there are several members here right now. And each of us is a subject matter expert doing different portions of an arcology or a habitat, a biological life support system. I'm the guts. And because several of these individuals either still have to present or went yesterday, I just want to sort of draw the larger picture on what everybody's position really is and why we have to think more holistically, with a W, holistically, really just cross-pollination and talking to others. So if I'm the guts, this is Pete. He's got one structural component system. His big thing is seasteading. And using plastic, waste plastic, turning it into plasticrete and building forms. And then I'm just going to be glossing over everybody here real quick, but just to give you an idea of, like, if the whole thing is a larger living structure, this would be the bones, one could say. Here's some of the things that Pete's working with. And I can send any of this later on. This is Frank, who went yesterday. Frank would be like the lungs. So he's building 3D printed systems for plant growth, among other things, but that's the most direct application for what we're doing. Some of his work there. So these things hanging inside of an arcology. There's Stuart, of course, and then Stuart is building also the bones. And so I believe he had presented some of this yesterday. I wasn't able to make that meeting. But just to give you an idea, more the structural components and the skin, if anything, the epidermis, the largest organ on our body. Some of the other people that he works with, Bill and James, and I'm not sure who the other one is there, but some of the guys over at the shop there in Livingston, and some of his structural components. Now, of course, then there's Breyer, who I work with as well. He's also another member. He would be the lizard brain, or the autonomic functions of the system. So, and not to get a whole lot of depth into this, because this is what he was presenting on yesterday, but this is how it works into what we're doing in a larger picture. So if we were designing our own spaceship, all of these components and systems would be required moving forward. And then back to me and my stuff. Okay, so just giving you some ideas. And now, of course, Dr. Tom Frida, who's after me, has also been joining us for our biology nights on the second Thursday of every month. And she'll, I don't want to take any of her thunder away, but then her specialty is the plant-based systems that are also enclosed life support systems. And then I would just be the mirror of that. So if plants, primary productivity, thinking back to trophism, I'm secondary productivity and all the other metabolisms that combined are still only 1/10 the efficiency of that primary productivity that she's mostly working in, but I'll let her speak for herself. Looks like I got a couple minutes left, so I'll leave it at that.
Great timing there, Colin. Do we have time for some questions, I think? No questions? Yeah, Stuart?
It happens.
Colin, I wanted to also mention briefly, or get you to talk a little bit about the food production using bioreactors. We had, in one of our bio nights, we had Novo Nutrients present to us. And so that's another application of a bioreactor that's very, very important for any environment, including the moon.
So they have I'm not sure if they're genetically modified organism. I believe they might have been, but don't quote me on that. I'm not sure. And they would be using another form of metabolism that I was still trying to wrap my head around it. That is producing essentially a high quality protein that is, I guess, more palatable or something along those lines, using a novel bioreactor system. So they would be the batch reactors, whereas I'm operating in a flow-through system. And of course, you wouldn't want to eat anything that's coming out of any of my systems unless it's like fish, something that can then be reprocessed again after the fact. You're certainly not going to go scraping the sides of one of these reactors and considering that, what do they call it, Vegemite or something. But yes,
I'll cover a little bit of that.
Great. Okay. But yes, Stuart, thank you very much. That is a good point. There are all sorts of applications for bioreactors, whether they be standalone or in a larger flow through system. And it's one of those things that I just don't think is given enough. It's given short shrift. But yeah, and that was another organization who presented for Arcology Solutions. So if you're interested, just send myself or Stuart or Nicholas, just a quick hello, and we can send you the link. It's, for people who are interested.
I have a bit of a question then. Is the main difference between your marshland in a box and the ordinary marshland that you try to make it go a bit slower, or could we just pour the things out in the marshland and they will have the same result?
There's a whole lot of nuance to that question. I think what you're asking is what's the difference between a natural wetland and its impacts versus a flow-through system like this with biological distillation. I guess it would be wasted opportunity. What you want is a homogeneous product versus a heterogeneous muck that you have to then reprocess. Like just I'll try to do it in half a minute. West Virginia University is taking mine drainage that's just a heterogeneous muck, and they're trying to re-separate all the rare earths out of that. So they're doing a stepwise pH adjustment process where they put everything back into solution with acid, and then they use caustic soda on 2 different steps to get rid of your aluminum and then your iron, so that they can finally get down to the manganese and the rare earths. You got to get the stuff, dewater it, move it, rewater it, acidify it, and then go through multiple steps versus getting it out in a biologically in a system potentially like this, where you just go right to your pay dirt right off the bat, your manganese and your rare earths, removing everything else. So that might be why you don't just. It's control. It gives you fine-tuned
controlled and structured wetland. So you know where you got the different parts of it.
Yeah. Thank you. Appreciate you guys.
Very good. Thank you.